Hi, this is Nolan Bentley with the Biology Spring Final Review. Congratulations on finally making it to the end of biology. So by the end of this video, hopefully, you know a little bit more and are ready for your final exam. Alright, so starting with number 1, we have the first mistake on the final. Number 1, it gives you obviously incomplete dominance if you end up completely unscrambling it. However, that is not the definition to the right. The definition to the right is when both alleles are dominant and they both show up, and that is something called codominance. So codominance is the answer. Had I said, were they blend? So a situation where when both alleles are present that they blend, that would be incomplete dominance. Okay, so moving on, we have the way an organism looks or its visible traits. And so that is what it physically looks like, or in other words, its phenotype. And the way to remember that is that PH sound at the front. Physical phenotypes. The genotypes, on the hand, which is the next question, are the actual genes you have. Genotype, yes. Alright. So, in order to create your genotypes, you first must replicate your cells, and then your parents' sex cells combined, and they made you, and then now you have your DNA, and you have to use that DNA. And the way we use all that DNA with all these genotypes in them is by turning them into protein. Now, it's this whole process of transcription and translation. But in the end, the things that are used to make proteins, or in other words, protein synthesis, are called genes. Okay, number 5, you have pea plants. They are spherical seeds, are dominant to dented seeds, which are recessive. It says in a genetic cross of the two plants that are heterozygous. Let's see if I can just steal this from messing everything up. A little bit more room. Close. Okay, there we go. So if we erase this, make this heterozygous. So we're going to make each parent heterozygous. That would require us to first consider what genes the parents could pass on. Well, they could pass on a capital S or a lowercase s. The other parent, if it's also heterozygous, could pass on a capital S or a lowercase s. Okay, so then you can mix those two sex cells. This is essentially what you've done. So like the ones I'm circling right now, we can pretend those are the mother's sex cells. These are the father's sex cells. When they combine, they make different combinations of children. Big S, little s, big S, little s, and a little s, little s. So what we have here are 3 different genotypes: homozygous dominant, heterozygous, and homozygous recessive. However, we only have 2 phenotypes, and you'll see the physical trait. You'll see that the question asks for the fraction of the offspring that have the spherical seeds. It does not care what genes you have, it cares what your overall phenotype is. So that would be this one, because it's got at least 1 copy of the dominant, this one, and this one, that all have a copy of the dominant. This is recessive, therefore, of those, 3 out of 4 children would be predicted to have spherical seeds. And so that is why this is your answer, 3/4. Had I asked what fraction would be heterozygous, for example, that would be 1/2 from this one and this one. But I asked spherical seeds only. Okay, so on here you're undoing one of those. You're just going backwards. What you can do is just kind of pick what's common in each column, and then what's common in each row. And you have to end up kind of separating them as you do this. So I'm going to work on the top first. I'm going to say that in this column, they both have capital S's. In this column, they also both have capital S's. So this parent must only have capital S's. On the other hand, the other parent, this row only has capital S's. This row, what it has in common is both capital S's and a lowercase s, but I've already used the capital S is up here. And so what I'm left with is that this parent must have donated that lowercase s. And thus you must have a homozygous dominant and a heterozygous parent making children. Number 7. It says list the possible genotypes for the following blood types. Okay, so blood type is one of those codominant genes. It's where when you have both genes, they are both expressed. So you don't just have A markers on your blood if you have AB blood type. You don't have a mixture of A and B type markers if you have AB blood. Instead, you have both the A marker and the B marker fully expressed and on separate places on the cell, and so you express both at the same time. The other example used in class for this was spotting. So like red and white spots on a cow. You don't get actually pink cows. You just get a mixture. Well, yeah. So there you go. You don't get a mixture. All right. For the following blood types, A type will be whenever you have the A gene and possibly another copy of the A gene, or if you have an A gene and a copy of the O. And the O is in the recessive version, and what we tend to signify that is with like lowercase I. It is recessive, therefore we use a lowercase. The I don't know what the I originally stands for. I like to pretend it's just an imaginary placeholder, because it's basically the lack of a gene is what it is. B will be very similar to A. It will be either 2 copies of the B gene, or 1 copy of the B gene and a lack of the B gene, or in other words, I. AB, on the other hand, is very simple. AB is AB. That's it. It is whenever you are heterozygous for both the A gene and the B gene. Let me bold these so they're a little bit easier to see. No boldness for you, I. Finally, the O type, you have to get 2 copies of that I version of the gene. And thus, lowercase I, resulting in a lack of either the A or the B marker on the surface of the blood. Okay, so we're going to use those ideas in this problem down here. Let me try copy and pasting that again. We'll make it a little bit smaller. That works, actually. Okay, I'll just put it over here. Cool. Let me copy that so I can do that in the future. Alright, so a woman with type A blood is claiming that a man with type AB blood is the father of her child, who is also type AB. So that her child is AB, we know at least one of the children will have type AB blood. Let me get a little bit more room then. Okay, we'll fix it when it happens. The mother has type A. However, if you look at number 7, that means we do not know what her second gene is. There's 2 possibilities. It could be another A or it could be an I. A man with type AB blood will have an A or a B gene to donate. So this child will have to end up having come from here, where the A and the B have combined. They could have had a type AA child. Could this man be the father? Well, we will see that that is a possibility with her current genotype. So, what we know is that, yes, it is possible. Sure, we don't know what other 2 types of children they could have, but we do know that one of the possibilities is A-type, homozygous A-type, and we know the other type is the heterozygous AB. There it is. Number 9. It says colorblindness is a trait that is found on the X chromosome. Now, that should tell you something. What that tells you is that this gene is sex-linked. That means it is on the sex chromosome, and thus it is affected by whether or not the child is a boy or a girl. So anytime we have sex-linked, one of the ways we can kind of simplify the process here is just by starting off by recognizing that, wrong way, that the parents will donate one of their sex cells. Girls will only be able to donate an X chromosome. Boys will donate either an X or a Y. What that tells us is that we can predict each of the 4 types of children they can have by mixing together their chromosomes. So they can have a girl. We'll fix that in a bit. They could have a second type of girl, depending upon which X is donated. They can have a boy, if the father donates his Y. And they could have a different type of boy if the other X from the mother mixes with the Y from the father. Okay, so a normal-sided woman, heterozygous for the trait. So the woman will be capital B, lowercase b. She marries a colorblind man, and so the man has a lowercase b and a Y, and you'll notice the Y doesn't actually have the gene. The reason why is because our Y gene is actually lacking a lot of information. It's very small. It lacks a lot of information that the X gene carries. So what is the probability that we will have a son, they will have a son that is colorblind? So if we look down here into the son category, so the girls don't really matter. In this in this Punnett square, the girls don't really matter. I can fill it out real quick just for completion's sake, although it's not really necessary because it doesn't ask about the girls. They can have a girl who's colorblind. That's cool. Oh, actually, that's the second part of the question. We'll come back to that then. For their boys, capital B or lowercase b. Okay, so what we have is of the son, a son who is colorblind. So if they have a son, of their possible sons, they have a 50% shot. They have a 50% chance that a son is colorblind. Notice I didn't say 25% because it didn't ask me what is the probability that that they will have a son who is colorblind. Actually, it's always unclear. If I'm looking up their possible sons, 50% is colorblind. So 50% of sons are colorblind. Of their possible children, 25% of their children will be boys who are colorblind. And then it says for a colorblind daughter, you will see that at the top right corner, they have a potential girl who is colorblind. So of their possible daughters, that's such a prettier name than sons. Daughters are colorblind. 50% of their daughters are colorblind. Of their possible children, 25% of their children will be girls who are also colorblind. So there you go. You'll notice that it's just very important how you ask the question. If you're ever confused on whether or not they're referring to all the children or if they're referring to just a section of the children, ask your teacher to clarify that, because that might just be a reading issue that can be very easily fixed. And no, I do not know which one it is on the test. I have not checked that out. So, like I said, ask your teacher. That'll help. Okay, so one sec. I am dealing with a little bit of lag here. Just give my computer a second to stop freaking out. There we go. It says, complete a Venn diagram comparing mitosis and meiosis. Alright, so mitosis is our body cells replicating. It's just how we make new body cells. So those are just normal types of cells. For meiosis, can I get it at the top of the... Yeah, there we go. For meiosis, it is our sex cells. I would say replicating, but really it's being produced from the cells that eventually become, that their offspring become our sex cells. So for both, though, they are both involved with replication. So I would definitely kind of put that as the link between those two ideas for them. Our replication of cells, or our divisions of cells. And technically, it's division of cells nuclei. The cytokinesis step is the division of the actual cell itself. Okay, so mitosis is the body cells replicating. There's a lot more information we ask you to know about this, though. It results in diploid cells. That it means the normal number of chromosomes. On the other hand, the meiosis process results in half. The number of chromosomes as usual, and that we call that haploid. So even though technically it means one number of chromosomes, it sounds like half. So that's really the easier way to remember it, is that it's half the normal number of chromosomes. Yep. Oh. For both have to separate DNA, of course. So have to separate chromosomes. So those are our strands of DNA. Alright, so what else do we need to know? We need to know that there is one division in mitosis. In meiosis, there are two divisions. For mitosis, mutations in them don't result in mutations in children. And therefore, for mitosis, since it's just your body cells, they actually don't matter as much in terms of mutations in your children. So, do not contribute to mutations in children. At least not very commonly. If maybe it was involved in the reproductive tract, maybe it'd have to be a very complicated process for mitosis to affect your children's DNA. For both column, though, our divisions of cell nuclei, that's the important part. Have separate chromosomes, important. I'm just going to call it at that. You can go check the key, see if anything else got added. So define diploid and haploid. You can just see the above. What it is haploid means the normal number of chromosomes. In humans, that is 46. Haploid means half the normal number of chromosomes. So in humans, that would be 23. They're significant because in our cells and all of your normal body cells, where mitosis occurs, you have 46 chromosomes, one from mom, one from dad. The exception of that is your red blood cells, which don't have DNA because they're special. Of your sex cells, on the other hand, you have half the normal number of chromosomes because you need to combine your DNA with a partner's DNA to make a child. So you only have 23, and it's a random mixture of your mom and your dad's chromosomes with a little bit of mixing between those individual strands as well. So you'll have mostly a mom chromosome for number 1, or maybe dad's for number 2, but they'll have little chunks of it, of the other parents on them. And that is from number 13, so we'll come back to that. What is produced by each of the above processes? Again, just see that body cells versus sex cells. If an organism diploid number is 20, what is the haploid number? That would be 10. Half that. Define crossing over. What process does it occur? Alright, so let's take that step at a time. Crossing over is during meiosis, when your mom and dad's DNA line up in the middle of the cell. Some exchanges occur of genes. Some exchanges of genes occur. This results in a greater mixing of DNA. Okay, so without this, the only mixing of your parents' DNA that your children would have would be whether or not they received gene number 1 from mom or dad, gene number 2 from mom or dad, gene number 3 from mom or dad, or from your mom and dad, so their grandparents. So this allows you to give each of your children a little bit of an extra mixture of your mom and dad's DNA, and we'll come back to that at the end of this question. It says, what process does it occur? That it would be metaphase 1. As the first metaphase, remember it does have 2 divisions, so there would be a second metaphase. So it's during meiosis stage is metaphase. There we go. Meiosis. So, DNA line up in the middle. This results in a greater mixing of DNA. What are the benefits of crossing over? The huge benefit is an increase in diversity. And so many things in our biology end up coming from that idea. We need diversity. That's why the U.S. is just absolutely a wonderful place. We have a huge amount of diversity, and diversity is essential to preventing populations from going extinct during times of change. And so all this diversity just basically allows the chance that a super baby will be born. That's right, super baby. They'll be really good at something. They'll do better than their parents because they came with just the right mixture of DNA, and thus they're very successful. That's what you're hoping every time you have babies, at least biologically speaking. Don't have babies. You're freshmen. Please don't. Your mom and dad's DNA. Cool. All right, so let's see if I can get this to work. I did practice earlier. Okay, so we're going to draw the stages of mitosis. So the first stage is one called prophase. During prophase, the DNA is just like all wiggly and whatnot up in here. It's starting to condense, and the nucleus is starting to disappear. So I'm not really sure how to show the nucleus disappearing. Can I make that a dotted line? I might. Dash type. Yes, I can. And there we go. Bam. Got it. Cool. Alright, so the nucleus is starting to disappear. By metaphase, it has now disappeared. This time, so somewhere in between here, you'll have crossing over starting to occur. And stuff like that. So let's do some more drawing. So during metaphase, the chromosomes line up in the middle. Now this is mitosis, so crossing over doesn't occur during mitosis. And let's draw the other side. So I'm drawing here mom and dad's version. So this is like dad's version of chromosome number, 2. Oh, no, I'm not. I'm sorry. That's just the replicated copy. So we had already actually replicated all the strands of DNA. And before this, during interphase, then they condensed. Now we have these structures, which are just this and this are copies of each other. They're joined at the middle so that during the next phase they can separate properly. So during the next phase, there's like these little structures that form called spindle fibers, and they pull them all apart. And when they do that, they give a very distinctive shape because they're pulling from the middle. And so they get like this little V shape. You usually can't see the spindle fibers too easily. In really good pictures you can, but obviously this is not one of those. And of course, probably the best thing you could go do is go search up a real picture. It's called mitosis. Just search mitosis picture, and what you'll see is steps that look like this. On the test, most likely what you're going to see is a picture of an actual cell. And so you have to be able to pick from an actual cell. Now, you know what I'm going to do for telophase, which is our last phase, is I'm just going to take this. I'm going to delete it. That's right. Just deleting it. I'm going to take this one. I'm going to copy it and move it over here, because basically telophase is just the opposite of prophase. The nucleus reforms, the DNA starts to spread back out, and so you stop seeing it all condensed into actual chromosomes, and it just separates back out into the strands of DNA. Anaphase, and finally telophase. However, the difference for telophase is that once we're at telophase, it's no longer just this one anymore. It's now two. We had two nuclei. I'll just stretch this cell, and in real life, yes, your cell just stretches. And there you go. Now you have two cells that can then divide in the middle. A process known as cytokinesis will cause that to happen, where it actually starts pinching in the middle, and the cells separate, their cytoplasm flows apart, and there you go, two new cells. What type of mutation would be transferred to an offspring? Well, what's above here is just mitosis, and during mitosis, I like to think of it as mitoses, because it happens in things like mitoses. My toes, get it? Mitoses, it doesn't matter. I can dip my toes in radioactive ooze and become a Ninja Turtle in only my toes, which is ridiculous, but we'll go with it. And that won't affect my children, assuming that, it doesn't transfer or something. That's because a mutation during mitosis has nothing to do with your sex cells. A mutation that is transferred to offspring must occur in your sex cells. So it must be two sex cells, possibly occurring, possibly caused by a mistake in meiosis. And that is going to be our most common cause during meiosis, because we do try to protect our DNA. That's why it's are buried inside of us. It's probably important to wear pants, stuff like that. All right, define non-disjunction and tell what effects it could have on an offspring. All right, so non-disjunction is, if you look up here, let's pretend that these guys actually failed to separate. So instead of separating properly, they actually grabbed and both went off to one direction. Okay, so that would mean like this is all lumped together right now. Okay, so just pretend a little bit. So if that happened, one cell, this cell over here will be lacking DNA. And over here, this side will have extra DNA. So it is, and let me undo what I just did so that doesn't get saved to my key. Okay, so define non-disjunction. When the DNA fails to separate. Get it? It fails. Non to separate would be to disjunction, which means to unjoin. We're not trying to trick you here. That's just how you break down the word. When the DNA fails to separate, it causes one of the cells to have less DNA than it should, and one of the cells to have more DNA than it should. And one thing to remember about the way our DNA works is it's really just like a stack of books, really, really, really tall. And if you put too many books on one side, it all starts leaning over and it falls over. Our DNA is this just wonderful balance that just so happens to work. And if you mess it up too much, it just doesn't work and the child won't develop. So whenever you have a non-disjunction, the vast majority of the time, the baby will never develop, and it's because it simply won't work well enough. It'll make too much of a protein if it has one more copy of DNA than it should. So it'll just make too much of a protein. That protein will accumulate and cause diseases, diseases like Down syndrome, Cry-du-Chie, not Cry-du-Chie, different one. But it'll cause diseases like Down syndrome where it just messes development up. On the other hand, if you have too little DNA, you most likely will die. There's only one that humans can survive, and that is whenever we have only one X chromosome. And that would, of course, be a girl, because it does not have a Y chromosome. And those girls will get what's called Turner syndrome, and they do okay. They have issues, but not so bad. Not as bad as they could be. So that's the only time we can survive that, because otherwise you're just completely missing parts of your DNA, and that's really bad. You're going to be making too little of certain proteins, and there's very few of those we survive. Well, only one that humans survive. So there we go. What causes mutations primarily? It is mistakes during replication. That is the huge main cause. Just mistakes made during replication of cells. I should say division. Division of cells. That could be a couple of things. That could be the replication of the DNA itself, or during meiosis. The other big thing is environmental factors. Most chemical factors really don't directly mess with your DNA. Most chemical factors mess with one of those two processes, replication of the DNA or meiosis, and they cause them to mess up. Of course, there are some environmental factors which are more interesting in this. Sunlight, for example, has certain types of wavelengths of light called ultraviolet radiation, and occasionally even things above that, but not too often on Earth. But ultraviolet radiation, or UV radiation, is able to actually break apart your DNA. And so whenever you're outside on a sunny day and that sunlight travels through your skin, because yes, your skin is not completely opaque, it hits the DNA of your cells and causes the DNA to break. And it is one of the reasons why people can get skin cancer. And the more susceptible you are to skin cancer is usually caused by lightness of your skin. It is definitely a factor that has affected our race. As we can tell, we're all very different shades of coloring, and it has to do with how much sunlight our ancestors were receiving. Okay, so define insertion, and we're talking about DNA here. The insertion of DNA is when extra DNA is added to a chromosome. We'll just say, there you go, when extra DNA is added. Substitution is when DNA is replaced with the same amount of DNA. And why that's significant, why there has to be this difference between them. First of all, just for clarity, but second of all, it does not result in a frame shift. It does not result in a frame shift, which is huge, and we will come back to that, I think. Let me go check, actually. A frame, of course, this is the moment my computer chooses to freeze. Okay, frame shift, not going to say what I actually typed, does not result in a frame shift. My wife just gave me a funny look. So define deletion. Deletion is when the DNA is removed, and so just imagine hitting delete on your keyboard. The DNA is removed, and therefore it's gone, it's missing, finite, done, and you're missing that information. That's a bad thing. Alright, so when DNA is missing, and that can often be a bad thing. Much like insertions, it can even result in frame shift mutations, which are very scary. So since that's not on here, let's define what that is. Can result in a frame shift, and whether or not it results in a frame shift actually has to do with how many are added or deleted, because our DNA is read in groups of 3. If it's added or deleted by a multiple of 3, it actually doesn't result in a frame shift, which is pretty sweet. But most of the time, let's face it, that's only every few numbers, so the majority of the time it will result in a frame shift. Can result in a frame shift, and let's define that where that is. That is when it can result in the DNA no longer being able to be read properly because the groupings of 3 are shifted over. And that is what we call a frame shift. And that is exactly as bad as it sounds. It means that ever after the insertion or the deletion event that causes a frame shift, it just doesn't work anymore. So yeah, that's bad. What must occur during interphase so that the daughter cells can be produced? A lot of things, obviously. There's a huge amount of stuff that we can say here, but it says interphase. And so interphase is specifically when the DNA is actually replicated. And this is a very common mistake. People think it's going to occur during meiosis or mitosis, but that is simply when the nuclei reproduce. Remember, our cells have like all these layers. Well, one of the insidemost layers, the nucleus, where our DNA is kept. And so since it's like stored there, it has to be broken apart, and that's this whole process. On the other hand, the whole cell also has to be divided. And so it's a much bigger process. So it's important to remember all the interphase and the cytokinesis as well. But anyways, DNA is replicated. That is the huge part. I mean, obviously, to make 2 children, you have to give each children something to run the cell off of, so you have to give them the DNA. I mean, if you don't give them something else, that probably isn't good, but the DNA is the big one. So what advantage do sexually reproducing organisms have over asexually reproducing one? I'm going to put this one in all caps because, dang, it's important. Diversity. We have a lot of drawbacks compared to asexually reproducing organisms. It takes us a while to find a mate. We have to develop the structures for sexual reproduction. We can't do it as fast. It just takes time. So asexually reproducing, much faster, much simpler, and less stuff can go wrong. A lot of things can go wrong during sexual reproduction, but they are cloning themselves. And what we get are new, let me get out caps, new combinations of DNA can possibly benefit offspring. And that's evolution right there, people. That's what it is. If you just so happen to have some traits that by mixing your mom and your dad's DNA, it is now more successful, you have evolved the human population. Remember, an individual can't evolve, but if you get to go make babies and then those babies inherit that trait, well, dang, you just started a population of humans that have a better trait. And so that's the idea of sexual reproduction. That's why we're always trying to find just the perfect match for ourselves, because our body desires to find somebody who our DNA can combine and make somebody awesome. That's what our bodies desire, at least. Take it how you will. Alright, so I'm going to stop here, go to part 2, if you need some more help. Alright, bye.